Hi everyone, this is Module 4, talking a little bit about soil microbial health testing. These videos are brought to you by On Farm, a program funded by the Canadian Agricultural Partnership, which is a federal, provincial, territorial initiative. When looking at soil health testing, the microbial aspect of soil is largely ignored. And that's not good because we know that the functions that the soil microorganisms carry out are crucial to soil health. Although there are some tests out there that attempt to measure soil health using biological indicators, there are some limitations to them that we'll talk about in the slides to come. It's really important that we develop a reliable and robust biological indicator. And again, this is something that is at the forefront of research. With new technologies coming, hopefully these tests are right around the corner. Soil health indicators need to be something that will change over time. We need to be able to quantify the changes in soil health. They also need to be measuring something specific, for example, the organic carbon content of soil or nutrient availabilities. And lastly, they must be able to be interpreted in a way that will help inform management practices, knowing if the indicator shows good or bad soil properties. These basic properties of soil indicators become challenging when looking at the soil microbial community. There are a lot of limitations that we need to be aware of when we're using microbial properties as a way to measure what's going on in the soil. The first one is that any soil sample you take is a snapshot in time. These communities change a lot through different weather conditions, whether or not a fertilizer or organic amendment was just applied, which crop was in the ground, and more. And the next is that they don't just differ across different conditions, they also differ across space. And I'm not talking about the top of a hill compared to the bottom of the hill. It's also vastly different in a crop row compared to between a crop row. There's no real benchmark or normal range to aim for in terms of the number of total microorganisms or the amount of diversity that should be present. So it's very hard to quantify what's good and what's bad just based on these metrics because it's so dependent on outside factors. And lastly, many microbial measurements lack clear interpretations. This just makes it hard to inform management practices. So looking back on what we've learned, we know that we can measure how the soil microbial community is changing based on the total amount of microorganisms present or their biomass, the actual activity of those microorganisms telling us how much are actually functioning in the soil, and the biodiversity of the community, which is looking at how many types of microbes are present. So some commercial tests already attempt to measure these metrics. The first one is phospholipid fatty acid testing, or PLFA. This is based on the idea that each microorganism in soil has characteristic molecules as their building blocks. PLFAs are one of those building blocks that can be used to define what type of microbes are present, also how many of each group are there, and how big the total microbial biomass is. Although this information is interesting, it's hard to interpret and make decisions based on the results. Specific biomass of bacteria and fungi can also be measured in many different ways, not just through PLFA testing. But the same problem arises. With no benchmark, this information is interesting, not all that helpful. One interpretation of these tests is to look at the fungal bacterial ratio. It's thought that having more fungi in your community, so a higher ratio, results in more effective nutrient cycling because fungi typically interact more with the plants. But this idea has not fully been proven. And to measure the activity of those soil microbes, you can measure the activity of microbial enzymes. All you need to know about enzymes is that it's something we can look for in the soil that tells us what the microbes were doing. The basis of this test is you can test for specific enzymes that you know are responsible for specific functions in the soil, usually carbon, nitrogen, and phosphorus cycling. The interpretation of this is that if there's a high presence of active enzymes, that targeted nutrient is limited. You can then use this information to inform further management practices. One problem here is that enzyme activity could also mean that there's an abundance of that nutrient present, making it really hard to predict what's actually happening. These indicators can be useful for tracking purposes as well. Noticing changes in these results over time alongside their soil health parameters is an indication that the community is changing with the management practices that's being put in place. That's why some important steps need to be taken when taking soil samples for these tests. Because so many factors influence the microbial community, your samples could return vastly different results that aren't due to changes in management. Taking samples in the same weather conditions, the same location in the field, preferably with the same crop in rotation, will be very helpful in trying to see changes that are happening in the community over time. So in research, we have the ability and the technology to get a full picture of the microbial community by looking at all of the DNA that's found in the soil samples. Seeing what specific microorganisms are present allows for the identification of all the functions that the community is capable of performing. It's less informative to look at individual species themselves, rather to group them by their function and look at what functions are dominating and which ones are limiting. These could involve getting a better understanding of groups of microbes that function to decompose organic matter, store carbon, cycle nitrogen and phosphorus, or ones that are really good at working together with the plant roots. Knowing these capabilities of soils will hopefully become an important tool for informing better management practices for soil health.